Hello everyone. Very good afternoon. I hope you guys had a great lunch, and then we are back to our bioequivalence studies. See, according to your syllabus, we need not talk about the studies that are conducted in bioequivalence or bioavailability. But then ultimately, you have to talk about the bioequivalence studies because the factors affecting the parameters that are evaluated for BAB are going to be the same. Okay, there are no much differences. So we have seen what all are the assessing factors for BA. They are going to be the same because once your ultimate aim of doing bioequivalence study is to find out the bioavailability is comparable or not, right? So the factors which are going to influence your bioavailability or affect your bioavailability are going to be the same for that of the bioequivalence. So by taking this opportunity when we are talking about bioequivalence, I just want to get you guys familiarized to the ideas that what do you mean by BAB trial designs, okay? Why do we conduct the BAB trials on human beings? What is the need? So by far we have seen that if the formulation is changed, if the strength of the drug is, if the ingredients of the drug are changed, if the manufacturing methods are completely changed, then definitely you'll have to go ahead and do the bioequivalence. When you want to compare the two formulation, that means one of them will be reference, one of them will be the study product. So reference could be the one which is already available in the market. If you want to do that, then you'll have to conduct the bioequivalence study. Then obviously for a modified formulation such as controlled release, prolonged release, etc., you'll have to go ahead and do the bioequivalence study. And obviously, of course, for intervention studies also, in order to check the drug-drug interaction and food effect of the food on the drugs bioavailability, that time also you'll go ahead and do the bioequivalence study. So these are the, and we have seen the various criteria also under which you'll have to conduct the bioequivalence study no matter what. In that criteria, we had seen study related क्या-क्या criteria रह सकते हैं and drug related क्या-क्या criteria रह सकते हैं, because of which we'll have to go ahead and conduct the bioequivalence study for a particular formulation, right? And when we are talking about bioequivalence studies, that means it could be in vivo or it could be in vitro. Okay, in vivo, why do we have to conduct the in vivo studies is because you have prolonged formulation, you have controlled formulation, then you have changes in the formulation, then you have changes in the manufacturing method, and you have in vitro bioequivalence studies. So there are two types of bioequivalence studies, in vivo and in vitro. Okay, so in vivo, in which you will be checking the bioequivalence inside a animal subject or inside human beings. Okay, so why you will be doing that? That criteria we have already seen. And in vitro जब भी कभी करते हैं, in vitro जब भी करते हैं bioequivalence studies, that time it is not necessary that you will have to conduct in vivo. Okay, that means the data which is there for in vitro should be sufficient enough for you to go ahead and put your drug into the market. And when this will happen, only it will happen only when you have the same drug, only the strength is different. ठीक है, मीन्स पहले हम 5 मिलीग्राम दे रहे थे तो थोड़ा डोज बढ़ा के हमने क्या कर दिया है 10 मिलीग्राम कर दिया. फॉर एग्जांपल इफ आई से amoxicillin tablets, antibiotic tablets, they are available in the market for 250 milligram and they are available in the market for 500 milligram as well. ठीक है, so only thing is that 500 milligram has to be taken once a day, 250 milligram has to be taken twice a day. So I need not conduct bioequivalence study on subjects for the same formulation जिसका सिर्फ strength change हुआ है, right? I have the in vitro data, ठीक है, so I'll be doing only in vitro data. That means I'll be doing only lab trial and getting the study results, and I will be getting that approved from the IRB or IEC because it's just the strength that has changed. And if there is any slight change in the formulation, only slight change in the formulation and slight change in the manufacturing, that time also only the in vitro data should be sufficient for me to go ahead and market my drug into the market, right? So those are in vitro, जहाँ पे सिर्फ in vitro data is sufficient enough for me to make sure that my drug is bioequivalent and I can go ahead and market it. Okay. And in vivo bioequivalence is something in which you'll have to do the in vivo trials. You'll have to do the animal trials as well. And you'll also, you might not have to do the animal, maybe not compulsory, but you'll have to either do animal trials or do subject trials and then go ahead and publish the data, get it approved, and then you can go ahead and do the marketing of your drug. Okay. So moving further in that area, we have designs of bioequivalence study. So क्या क्या designs हम रख सकते हैं in bioequivalence studies? Designs of the bioequivalence studies are not going to be different than that of the clinical study designs and types that we have studied. They are going to be the same. Okay. But what all are the preferred methods, या फिर preferred types of design that are used in bioequivalence study? So the test and reference drug formulation must contain, ठीक है. First is pharmaceutically equivalent drug, ठीक है, in the same dose strength. So अगर तुम test and reference drug ले रहे हो तो सबसे पहले वो pharmaceutically equivalent होने चाहिए. In the sense the active drug should be same, the excipient should be same, everything should be same. ठीक है. Only the thing is manufacturer is different, the brand name is different. Okay. The same dose strength should be same. It is in similar dosage, for example immediate release or controlled release, तो दोनों भी immediate release होने चाहिए या दोनों भी controlled release होने चाहिए. Given by the same route of administration, so you cannot take one is given orally and another one is given parenterally. You cannot do that. Then you have to have approval from IRB of the testing unit. Then it consists of both single dose and multiple dose studies. Okay. So these are the things that your test and reference drug should be conducting in order to for you to go ahead and conduct the bioequivalent trial on your drug with the help of a reference drug. Okay. So these are the things that you have to have in place in order to in order to start the bioequivalence trial. Okay. So BE trial, it is called as BABE trial. Then elements of a bioavailability study protocol, bioavailability or bioequivalence protocol. So generally we call it as BABE study protocol. Okay. So it is same as that of the clinical protocol that we have seen by far. Okay, so it has title, then you have principal investigator who is the study director, then you have project or protocol number and date. Okay. Then you have study objective, then you have study design. In design, what kind of a design you have, then you have drug product test and reference, uski information rahegi. Then you have dosage regime, then you have sample collection schedule. Sample collection in the sense we are talking about what is your blood collection time points. Okay. Then housing and confinement, how are you housing the people, how many people, fasting and meal schedule, very important. जैसे meal schedule, जैसे मैंने बोला था, we have to keep a track of everything, that when that person, the subject who is in your facility, when that person went what time he ate, okay, how much of calories he ate, what time he had lunch, what time he had evening snacks, and what time he had dinner. So all this data is kept. Okay, because you'll be checking, because what are you, by doing this, what are you trying to do is you are trying to keep all the other variables constant, so that whatever data you'll be getting, it will be pure the study output. Okay. Then analytical methods, what kind of analytical methods you are going to use in order to evaluate the blood samples or urine samples. Then study populations, the subject selection, medical history, physical examination, laboratory test, inclusion exclusion criteria, details of those criteria, restriction and prohibition. So restriction and prohibition में क्या आता है, अल्कोहल नहीं लेना है, निकोटीन नहीं लेना है, means you cannot chew tobacco, cigarettes, etc. So generally that point comes into restriction and prohibition. Then clinical procedures क्या follow कर रहे हैं, dosage and drug administration कभी हो रहा है, then biological sampling schedule कभी होगा, handling procedures क्या रहेगा biological samples का. That means once you remove the blood, once you take the samples from the subjects, how are you going to handle it, where are you going to store it, under what condition, how are you going to transfer the samples to other facility, all that has to be mentioned. Then activity of subjects, then ethical considerations such as basic principles of good clinical practices are there, then institutional review board, informed consent, indications of subject withdrawal, adverse reaction and emergency procedure. What are other facilities? Then data analysis will be having analytical validation procedures, statistical treatment of the data, and then drug accountability and appendix. So if you see this protocol, it exactly looks similar as that of the clinical trial protocol. Because over here also we'll be checking on all the aspects which are required from ethical point of view and evaluation kind of view. Okay? So everything that comes into picture with respect to ethics and everything that comes into picture with respect to evaluation of your samples that you have collected from the subjects will be taken care of. And everything has to be the same as that of a clinical trial. Okay, so this is how the protocol of template would look like. Okay, then analytical methods must be accurate. Okay, of sufficient sensitivity to measure small changes. They should be with appropriate precision. Then measure the actual concentration of active drug or active metabolite achieved in the body. So your analytical methods should be in place basically. Then reference standard. What is reference standard over here? So one is like a reference drug that you're going to use. One formulation of the drug is chosen as reference standard against which all other formulation of the drug are compared. So reference drugs should be administered by the same route as other drug formulations. Reference standard is generally a formulation currently marketed with a fully approved NDA. So this is a very crucial requirement for the reference standard that you're going to use for your BAB trial, because it has to be fully approved, for which there are valid scientific safety and efficacy data. Okay? That kind of a reference standard only you can use for conducting a trial. And usually innovators or brand drug. So generally it's a brand drug. ठीक है, अगर मैंने कुछ मैन्युफैक्चर किया है तो I'll be selecting reference drug as the best formulation which is there in the market for my drug. Means like in the sense, if I have manufactured diclofenac sodium, then I will use the diclofenac sodium reference standard which is manufactured by Cipla या फिर GlaxoSmithKline, something like that. So I'll be taking a brand drug only for my reference standard. Then extended release formulations, they are done in order to ensure that the product has claimed controlled release characters. No occurrence of dose dumping. What do you mean by dose dumping? In the sense that once in controlled extended release formulation, generally how they are formulated is that you have a higher dose, okay, and the formulation is made in such a way that at some time interval in 3 hours maybe it will release this much amount of, say 10 milligram of the drug. In next 3 hours it will release another 10 milligram of the drug. But when first time जब उसने 10 milligram drug release किया, that time it has eliminated also, means after 3 hours it will be getting eliminated. So dose dumping नहीं होना चाहिए, in the sense that when the drug is getting released, okay, it is not getting eliminated out of your body. That means 10 milligram release हुआ, but it is not getting leaving your body. Then another 10 milligram release हुआ, but it is not eliminated from your body. So what is happening? The dose is getting dumped inside your body itself. So that means the dose is getting the dose strength is getting built up in your body. Sorry. So that is called as dose dumping, when the elimination process is not taking place properly. So you have to ultimately change your formulation, make sure that what is the goal of your extended release formulation is to release the formulation for an extended period of time. Okay? So that can take care of the therapeutic effect that you are willing to achieve. Okay, so that dose dumping should not happen, means that the dose should not get saturated inside the body of a person. Okay, so in order to tackle that, generally bioequivalence studies are done. So you have to, by doing this bioequivalence study, you will be making sure. Why we are talking about controlled release and extended release formulation only for BAB? Because these are the formulations of which the active drugs which we have are already available in the market, या फिर already are getting sold out in the market by for 2 dose a day, 3 dose a day, something like that. अगर मुझे किसी ने बोला कि तुम्हें दिन में 3 फीवर के टैबलेट खाने के बजाय अगर एक ही टैबलेट तुम खाओगे तो तुम्हें दिन भर में बुखार बिल्कुल भी नहीं आएगा, then I would prefer eating one tablet, right? So, but then see, now I will not be going ahead and doing the clinical trial for this modified formulations, because उसकी जरूरत नहीं है, क्योंकि active ingredient पे already clinical trial हो गया है. The only thing I have to check is that bioavailability and bioequivalence are in place or not. ठीक है? So that is when I'll be doing bioequivalence studies and not doing clinical trial. Why? Because the only thing that is modified is your dosage form is modified. Okay, so active drug is not modified. When the active drug is different, that is when you will be doing a clinical trial, not BAB trial. Okay, so steady state is equivalent to currently marketed extended release formulation. You will check this point also by performing a BAB trial on extended release formulation. Then consistent pharmacokinetic performance between the units. New extended release product compared with the existing non-controlled release product. Okay, so non-control in the sense you will be say, for example, I'll be having one paracetamol tablet which I'll be comparing it with the new extended release paracetamol tablet. Okay, so what is my end goal? That both of them should have the bioavailability that I am looking forward to. ठीक है, नॉन कंट्रोल्ड रिलीज प्रोडक्ट इन द सेंस उसका बायो अवेलेबिलिटी भी कुछ रहेगा ना बॉडी में. फॉर एग्जांपल 100% है एक डोज खाने के बाद. So my new extended release product should also be able to give that much of bioavailability. If it is not able to give that much of bioavailability, then the product is not good enough for me. Okay. So next is combination drug products is to determine the rate and extent of absorption of each active ingredient and to determine if it is equivalent to the rate and extent of absorption of each active ingredient administered concurrently as separate single ingredient preparation. So basically you'll be giving 2 drugs at the same time. ठीक है, so वो individually drugs का clinical trial शायद हो गया रहेगा, but now you want to do what? In clinical trial, everything is safe, means that there is no side effect, no toxicity data, no. So the efficacy and the safety has been evaluated. Now the only thing you want to do is, if I give these 2 formulations together, are there any drug-drug interaction? So those kind of studies will be going under the BAB trials. Okay. Next is, so this is how the design of the protocol looks, and how you have to take care of the analytical methods in the BAB trial, and also what do you mean by reference standard, and what do you mean by extended release formulation BAB trials, and what do you mean by combination drug product BAB trials. That is what they have to. Then what do you mean by study design? Okay. Then fasting study, food intervention study, and multiple dose steady state study. Okay. So fasting study and food intervention study. This fasting study and food intervention study I have already talked to you guys about. Still we'll go ahead. So fasting study done for immediate release and modified release oral dosage. Okay. So male, female subjects may be used. You can use any subjects. Then blood sampling is done at appropriate interval to obtain the plasma drug concentration versus time profile. Then subjects should be in fasting condition at least 10 hours before drug administration and 4 hours after administration of the drug. So this is how the fasting is maintained. So when I was in Glenmark, most of the time for a single drug, ठीक है. We used to do fasting also and fed study also. ठीक है. So generally we used to do it for immediate release and modified release. So now think in this way. See, what is the need of the time currently? Innovation is the need of the time. Okay, so we have, how will you reduce the work of doctors and how will you reduce the confusion of the patients? That is what the companies are trying to, pharmaceutical companies are thinking of. Okay, so when they use an immediate release or when they make a modified release oral dosage forms, they have put a lot of thought process in creating those dosage forms. ठीक है. So I am creating a dosage form for a drug which is already available in the market. Modified, I'm creating a modified dosage form for a drug which is already available in the market. Why I am doing so? I'm doing so to achieve some good results out of that drug. So if I make a drug into modified dosage form, for example, say I made a immediate release insulin tablet. I'm just saying insulin tablet. There is no insulin tablet in the market. Insulin is given only in the injection. So, but if I make a insulin release tablet, so that means immediate release. That means if I have to give it to a diabetic person, he'll have the effect in within few hours or maybe half an hour or within few minutes. So that is an achievement. For that, I do not have to go ahead and do the clinical trial. Okay, I'll just go ahead and do the BAB trial in both the by doing either fasting trial, by doing fed trial. So I'll just check these 3 points. I'll just check these 3 points: fasting study, food intervention koi hai kya, and if I do the multiple dosing, how will I achieve the steady state? Okay, so that is what I will be looking into. So why I'm repeating these points is to just to let you know that there is a difference between the clinical trial and BAB trial, and why BAB trials are different than that of the clinical trial. So clinical trial is basically done for everything that is new. And BAB trials are generally done for a modified formulation, or just when you want to check that whether the drug I have manufactured is good enough as that of the drug which is available in the market or not. Okay. So I'm making myself clear over here. Yes or no? Are you guys understanding? Shabdali, Vaishnavi? Where the people I ask, they never write. Shabdali, are you there? Yes. So this was about the fasting study. Okay. Then food intervention study. So studies are conducted, and yeah, let me tell you about this point also over here. Subject should be in fasting condition at least 10 hours before. So that means you have to make sure that the subjects who are there in your facility are not even drinking water for 10 hours. So they'll be sleeping basically. अगर मुझे सुबह 8 बजे उनको डोजिंग देना है, अगर मुझे 10 बजे सुबह उनको 8 बजे उनको टैबलेट देना है, that means 10 hours before that they should not be doing anything. And I tell you, at when I am telling you guys over here, it is very easy, sounds good, fun. It's like, yeah, this is the protocol, but then subjects who are there in the facility, they do so much of naughty that they cannot survive without water. They have to drink water no matter what. But then, see, you have to step down and do something, because if you change the protocol little bit also, you have to, your readings will be changed. Okay, so these variables have to be constant. ठीक है. And after this also, dosing also 4 घंटा उनको पानी नहीं देने का है. That means it's a difficult task for subjects also to do it, because almost for 14 hours they are going to have nothing, no food, no water. So, and that is actually followed, huh? It is not only on the paper that we have written over here. This has to be followed. Okay, so it's not a easy task to become a subject also. Then food intervention. So studies, intervention studies. These studies are conducted after high fat and high calorie meal. Okay. Subject should be in fasting condition at least 10 hours before drug administration. Meal is given 30 minutes before the dosing. Okay. So 30 minutes before dosing, we'll be giving them high fat, high calorie meal. Okay. No food given for at least 4 hours after the administration of the drug. Done for modified release dosage forms and for immediate release form if the bioavailability is affected by the food or not, generally for ibuprofen and naproxen. So these are the examples of the drug for which we do it. Okay. So the protocol is the same, and exactly I have seen following the same protocol in BAB trial at Glenmark facility when I was working there. So definitely high fat calorie basis, they literally, we used to give them cheese sandwiches and etc. to make sure the calorie count also comes, calorie count also is been written in the protocol. So कितना कैलोरीज का ब्रेकफास्ट देना है, 1500 कैलोरीज का ब्रेकफास्ट, 2000 कैलोरीज का ब्रेकफास्ट, वो भी मेंशन होके आता है प्रोटोकॉल का. ऐसे हम खुद के मन का नहीं देते कि इसको एक सैंडविच, इसको 2 सैंडविच. So accordingly the calorie count is also been notified in the protocol, and those calories only, that much calories only subject is supposed to eat. And then after he will be given the dose, 30 minutes after he will be given the dose, and then 4 hours he'll be again fasting himself, and then they will be provided with the lunch, dinner, snacks, etc. So that is how it is. So that basically you'll be checking in this study, you'll be checking the effect of the food on the drug. Okay. Then multiple dose steady state study done for oral extended controlled release drug products, and done in addition to fasting and food intervention study. 3 consecutive C minimum measured on 3 consecutive days to determine the steady state. So 3 consecutive C minimum means concentration, minimum concentrations will be measured consecutively in 3 days to determine the steady state of the multiple dose that you have given of that particular drug. Then sampling done similar to the fasting study. So sampling in the sense the blood withdrawal part will be the same as that of the fasting study. So basically in multiple dose trial, you will be checking that what is the concentration level, the steady state of the concentration of the drug that you are achieving into the bloodstream. Okay. So that is what will be checked. So along with, for a particular drug, along with the food, fasting, food study, and you can also perform the multiple dose steady state trial. Okay. So this was it. Then next is crossover design. So generally for BAB, crossover design is very famous. We already know what do you mean by crossover design. A and B agar treatment hai, so in the second period, the first person will be getting A and second person will be getting B. So vice versa. It depends on that. So each subject receive the test and reference drug product. So both of them will be receiving it either in period 1 or period 2, whatever. Example of this, how the Latin square crossover design is the statistical method through which we determine which subject will be getting which drug in which period. Okay? So basically it's a statistical design which is invented for the randomization of the subject in the crossover design. Each subject receive each drug product only once. Adequate washout period is provided between the drug. Okay. Advantages, subject to subject variation is reduced. Okay. All patients do not receive same drug product on the same day. So that is also one advantage. So that you can make sure that ये drug से न्यू मेरा जो मैन्युफैक्चर ड्रग है उससे कोई साइड इफेक्ट है तो मुझे पहले ही पता लग जाएगा. I don't have to wait for another time. Okay. So this is how the Latin square crossover design look like. So you have subjects number. This is study period 1. So subject number 1 is receiving drug A in the first period, then receiving the drug B in the second period, and receiving the drug C in the third period. So this is for 3 drugs that you're in. So either it could be a placebo, standard, and the study treatment. It could be like that. Okay. So in between this period, just a minute. So in between this study period 1 and study period 2, this is what is our washout period. Okay. And this will be also our washout period. Okay. So this is how the Latin square design will actually look like, and this is how the document actually looks like, which comes to us for the after randomization. And then next is so if you have four study of 4 drugs products in 16 human volunteers. So this is how the randomization will look like, Latin square crossover design. Okay. So basically, you are making sure that each subject is receiving each treatment in this 4 period of time. Okay. So period is what? It's a time period in which the study is performed. Okay. So when I'm saying period 1, that means 3 days for period 1, 3 days. So it will start from the time subject has enrolled into my facility for the BAB trial. Okay. Until I withdraw the last blood sample from him. Okay. So that would be one study period. After that, he'll be going home for washout period. So that washout period is generally of 15 to 20 days. And then again, the same cycle is followed until we reach to the period 4. So 2-period study performed on 2 different days separated by washout period, generally about 10 elimination half-lives. Okay. Uh, 10 elimination half-lives in the sense, maybe I can explain this in the next this thing, but elimination half-life in the sense that you will be checking the you should be knowing that what is the elimination rate of the drug and half-lives of the elimination of the drug, in the sense half of the drug is getting eliminated at what time point. Then अगर 50% eliminate हो गया already, that means 50% already body में बाकी है, तो 50% में से 50% कभी eliminate होगा, so that will be another half life, the elimination half life. So you have to make sure that 10 times you have counted those elimination half lives, and those many days, वो 10 elimination half lives achieve करने के लिए कितने दिन लगेंगे, उतने दिन का washout period cover होना चाहिए. Understanding? So this is how the washing period is decided. Then sequence, number of different orders in the treatment group is in a study. For example, 2 sequences, 2-period study would be designed as follows. Sequence 1, T and R, that is test and reference. Sequence 2, reference and test. Okay. So agar 2-period study hai, in sequence 1, few people will be getting treatment, few people will be getting reference. And in sequence 2, few people will be getting reference and few people will be getting test medication. Evaluation data, it will have analytical methods mentioned in it, and also pharmacokinetic evaluation of the data, that means Tmax, Cmax. You will be using software such as ANOVA, which is a very popular software for biostatistics, in biostatistics for evaluation of the study data. So that all will be used, 2-sided, one-sided test procedure. So that all depends on how the protocol is designed. Okay. So evaluation of data I'm not going to talk about, because this generally is biostatistical part. When we study about biostatistics, that time we can look into it. Okay. So now there is only less than 1 minute remaining. If there are any doubts, you guys can tell me. Are there any doubts? If not, we can end the meeting. Are we fine till here? Okay. Okay, then I'll see you off for the day, and we'll see you directly on Monday.